So our project was on the effect of salt concentration on different invertebrates. The purpose was to determine whether or not salt concentration in water has an effect on the resting membrane potential of freshwater crayfish or blue spiny lobster, as shown here.
Invertebrate will be changed by manipulation of the Goldman-Hodgkin-Katz equation. Resting membrane potential is controlled by two factors, ion concentrations and ionic permeability. This is important because it can help us advance our knowledge of resting membrane potential in other living organisms as well.
Our hypothesis was that as the salt concentration was increased, the resting membrane potential of both invertebrates would become more negative.
So for this dissection, we used one freshwater crayfish and one blue spiny lobster. They were placed on ice for 10 minutes for a natural anesthetic so that no pain was felt by the animal when the tail was cut off. 3 saltwater concentrations were made for each animal, including brackish, seawater, and hypersaline solutions. These were made using table salt, 4, 7, and 10 grams in that order. The dissection was performed to reveal the superficial muscle fibers of the crayfish and the blue spiny lobster. The micromanipulator and intracellular microelectrode were lowered into the invertebrate to measure resting membrane potential in the neuroscience lab in the SCAP Science Center.
In order to create the desired salt concentration, table salt was measured out using an N-tris scale balance and then was added to a 200 milliliter of tap water and mixed using a magnetic stir bar until completely dissolved for all 3 of the solutions. The brackish solution contained 4 grams of salt and 200 milliliters of water. Seawater was 7 grams of salt and 200 milliliters of water, and the hypersaline solution was 10 grams of salt and 200 milliliters of water.
These are our dissection images. As you can see, the crayfish tail is on the left and the blue spiny lobster tail is on the right. So they both had swimmerets. Unfortunately, this picture doesn't show, but the swimmerets were cut off before the dissection was performed.
So the first step in dissection is to cut the tail off of the invertebrate being dissected. We did do this one at a time, first with the crayfish that had been sitting on ice for 10 minutes as a natural anesthetic in order to follow the animal policies of Hope College in research. The tail was then pinned down to a Siligard dish and filled with the control normal crayfish solution. The swimmerets were then removed, as you can see in the slide before. On the blue spiny lobster tail, these feather-like things are the swimmerets. They have been removed in this picture on the crayfish tail in order to reveal the superficial muscle fibers. After the swimmerets were removed, a deep incision was made between the sternites using a scalpel. The sternite lies along the middle and is the bony structure, as you can see right here. The cuticle was then completely removed using scissors and tweezers. Now, this is a longer process as an incision is made along the bottom, being careful not to cut any of the superficial muscle fibers, and the cuticle does have several layers to cut through, so this can be a tedious process in order to preserve the muscle fibers used in the experiment. The superficial muscle fibers that were used were revealed on both invertebrates in order to use the microelectrode in order to measure resting membrane potential.
The many tests performed. A correlation analysis was done for both the crayfish and lobster to see how increased salt concentration affected the 2 invertebrates individually. A scatter plot was made for both to show this correlation. A pre-post t-test was done to compare the first controlled normal saline to the 5th normal saline solution in both crayfish and the lobster individually. This was to see if salt concentration or time had an impact on the invertebrates. Bar graphs were made to show these results of these pre and post tests. Test continued.
Next slide shows the other tests that were used. The lobster and crayfish were then compared at each level of salt concentration, not including the 5th control solution used for the pre and post test. Therefore, there were 4 more t-tests and bar graphs created comparing the crayfish to the lobster at each level, comparing normal saline of the lobster to normal saline of the crayfish, brackish versus brackish, seawater versus seawater, and hypersaline solution versus hypersaline solution. These were ran at the concentrations of 0.005 for the normal saline solution, as you can see here in the first place in the scatter plot, and then 0.342 molar for the brackish solution, 0.598 for the seawater solution, and 0.855 molarity for the hypersaline solution. Now, normal solution for crayfish would be right about here for freshwater, whereas in a blue spiny lobster, the seawater concentration of 0.598 is what would be considered normal. As you can see here on the scatter plot for the crayfish, that they are relatively similar, that there is not a lot of overlap between the saline and the brackish concentration, but there is some between saline, seawater, and hypersaline for the freshwater crayfish salt concentration. This is a scatter plot for the blue spiny lobster. As you can see, it has relatively similarity in looks between the scatter plot for the blue spiny lobster and the scatter plot for the crayfish. These were done at the same concentrations for salt, including normal saline, brackish, seawater, and hypersaline. These solutions were, as stated previously, made using 4 grams of salt, 7 grams of salt, and then 10 grams of salt mixed into 200 milliliters of water. As you can see, there's a little bit of overlap between the seawater and brackish concentration, but not the hypersaline or saline solution. So these are the results for the crayfish for the pre-test and post-test, which includes the normal saline solution in solution number 1, and then the saline solution once again used as a control as the 5th test. This was done to see if there was any decay over time included as a factor in our experiment. The salt concentration of the water did not show significance in average resting membrane potential for the difference in any solutions for the freshwater or crayfish. As you can see here, for the normal saline solution, the p-value for this test was greater than 0.05, so there is no statistical significance there. And the error bars are overlapping for mean resting membrane potential in the crayfish. Now, the same results were also considered for the blue spiny lobster. The salt concentration of the water did not show enough significance in average resting membrane potential difference for the pre-test and post-test solutions of normal saline for the blue spiny lobster. This had a p-value greater than 0.05, so again, no statistical significance there. So for the comparison of the results between the two invertebrates, we had a comparison bar graph showing the crayfish in the light blue and the blue spiny lobster in the dark blue between all solutions. Unfortunately, there were no significant findings in the difference in resting membrane potential between the blue spiny lobster and the freshwater crayfish. All the t-tests and correlational analyses lied above the range of statistical significance with a p-value greater than 0.05. There are a few that were close, but none that ranged in a 95% confidence interval. Now, this did not support our hypothesis that the blue spiny lobster would have a better more average resting membrane potential for seawater, but as you can see, the more salt concentration that there was, the closer the error bars got and the less negative they did over time. So these results signified that as of now, there is no significant finding in the difference in resting membrane potential in different salt concentrations or between the freshwater crayfish and the blue spiny lobster. The value of this work is that later studies could be published or research could be done with a higher salt level concentration to see if there's any significance there. Some sources of error could include the dissection done that's cuts the superficial muscle fibers, not letting the invertebrate rest in the solution for long enough to acclimate to that environment. Future research could also look into studies that can be repeated using different species of invertebrates, such as other lobster species or different types of organisms that are found in seawater or brackish or freshwater concentrations like fish or other mammals. This was just done on invertebrates, so we followed the animal cruelty policies that the college provided. All right, any questions?